one. We're talking about neuronal integration, where you could either have two signals come in really quickly from the same axon, that's temporal summation in time, and if those add together to reach threshold, we get the axon firing. And we can also have spatial summation, where more than one axon is sending a signal to the dendrites at the same time, and they get added together. Okay? And I didn't talk about these guys over here on the side, but it's a pretty fun way of thinking about what we're looking at. Okay, so in no summation, it's like this axon is sitting here casually shouting fire every few minutes. You're probably not going to do anything. If they're just going fire, fire, you're probably just going to sit there and look at them like they're crazy. Okay? And so that's what happens with this first one. We don't get an action potential. In temporal summation, there's one person who shouts fire quickly over and over and over and over again. That's going to get you excited enough to run away or cause an action potential. Okay, so that is temporal summation. Spatial summation is where you have many people shouting fire at the same time. That's also going to get you excited enough to run away. Okay, so you're also going to cause an action potential. Then in spatial summation of your ESPSs and EPSPs and IPSPs, sorry, messed that up. So you get an excitatory signal and an inhibitory signal at the same time. That's where one guy's yelling fire, one guy's yelling, oh, it's not a fire, no problem, just stay here. And you have to weigh out who is speaking louder and what should you do in that instance. Okay? So that's where we were at. That's how this picture comes in of temporal summation. So temporal summation in time, here are your excitatory stimuli. They happen quickly enough. They all add up together to eventually reach that threshold, which causes the action potential to fire. All right. So far we've looked at, in our big picture we drew up there with the moose-looking neuron, we've looked at the information coming into those dendrites or to the soma. And then the information gets integrated there at the axon hillock, and you decide whether to do an action potential or not. Now, this next slide is looking at presynaptic inhibition. So this is after all of that has happened, and we've sent an action potential all the way down that axon. There are some instances where there is an inhibitory neuron that innervates on that postsynaptic neuron right at the synapse. So this is the long axon. The action potential has made it all the way down here, and he's ready to release the neurotransmitter. So he's ready to open calcium channels, calcium will come in, the neurotransmitter will be released. Okay? But because this inhibitory neuron is here, if he fires, he releases his own neurotransmitter, which is enough to inhibit that action potential that's traveled all the way down that axon already. So he can put the brakes on the firing of this axon right at the very end. Okay? So that is presynaptic inhibition right there at the synapse. We have the ability to inhibit an action potential so the calcium channels aren't open and we don't actually release our neurotransmitter. Okay? But it's just happening at a little bit different location. All right. Speaking of neurotransmitters, more than 100 different chemicals have been identified as neurotransmitters. Now, luckily for you, we don't expect you to learn all these neurotransmitters. We just want you to know what a neurotransmitter is. Okay? So know that it's released by that presynaptic neuron. It goes into the synaptic cleft, and it binds to a receptor on the postsynaptic neuron. Okay? That is what a neurotransmitter is. And it's going to bind to receptors that are ligand-gated channels. So they're going to open and close. And they're going to allow certain things through. Now, some of these you've already seen before, like acetylcholine. Okay? We know that acetylcholine is the neurotransmitter used for skeletal muscles. Is acetylcholine an excitatory or an inhibitory neurotransmitter when we're looking at skeletal muscle cells? Does it excite them or inhibit them? It excites them, right? Because it tells that muscle cell to contract. Okay? So in skeletal muscle, acetylcholine is an excitatory neurotransmitter. Okay? If we look at cardiac muscle, though, that same neurotransmitter inhibits the activity of a cardiac muscle. So it just depends on where we're releasing these neurotransmitters will determine what effect they have on the postsynaptic neuron or that effector that it's going to. Okay? Some other ones here you might have seen before, epinephrine and norepinephrine, so adrenaline with your fight or flight response. Okay, so that's there. Serotonin, histamine, dopamine. Dopamine is your natural painkiller. Okay, substance P over here is your pain-related neurotransmitter. Okay, so we have lots of different neurotransmitters. Again, you don't have to know them. We'll only talk about specific neurotransmitters as they relate to specific organs, like acetylcholine. Okay. All right, finishing this chapter up, we're going to talk about regeneration of nerve fibers. So we've already said that most of our neurons are amitotic, so they don't divide. But if there is an injury to that neuron itself, it may be able to regenerate like an axon that's been broken. Okay? So the peripheral nerve fibers can sometimes regenerate if the cell body, the soma, isn't damaged. And some of the neurolemma remains intact. So remember, the neurolemma is that exterior region of the Schwann cell. So we're leaving, well, I'll have a picture on the next slide. We're leaving the tube of that Schwann cell, that myelination, but the axon that ran through that myelination is gotten rid of. Okay, so if we leave the tubes, that axon can grow back through the tubes and reconnect. Okay, so that's what the next bullet said. The neurolemma forms what we call a regeneration tube, through which the growing axon can reestablish its original connection. If the nerve originally led to a skeletal muscle, then while that nerve was not connected, the muscle can atrophy, can break down. Okay, because it's not being used. But when that neuron regenerates, you may be able to build that muscle back up over time through some physical therapy. And currently there's a lot of research going into this area to try to figure out how we can make nerves grow. Because if you have a major damage somewhere in the pathway, let's see if I can draw a picture for us here. I'm not good at pictures, so we'll see what happens. Okay, so if we look at the brain and the spinal cord, and there's lots of nerves that come off here to feed to your arms and your legs and everywhere else. Both information coming in to the brain, sensory information and motor information going out. If we have damage in certain areas, it's going to block off everything beyond that area. So, for instance, if we have damage in the cervical area of your spinal cord and we sever that spinal cord, that cuts off connection from the brain to everywhere else. So sensory information won't be able to get up to the brain, and motor information can't get from the brain down to the rest of the body. So depending on where your injury is in the spinal cord, you could become a paraplegic, a quadriplegic, because you're losing nervous system information to a whole area of the body. Okay? So that's where things like stem cell research comes in. Try to put stem cells into that area so that the area can regrow. I feel like there was one more slide for that PowerPoint, but if there wasn't, it's okay. No. Okay. Good, good. Then that means chapter 12. Yay, the central nervous system. Central nervous system. I brought my thinking cap with me. So when we talk about the central nervous system, we're talking about the brain and the spinal cord. Okay? We're going to start with the brain, and then we'll get to the spinal cord later at the end of chapter 12. Okay? So just to get us introduced to the brain, I know that you guys have done your sheet brain dissection, so some of this should be review or at least remind you of what you've talked about already. Okay? So first of all, brain size is proportional to body size. So the bigger your body, the bigger your brain. The smaller your body, the smaller your brain. Okay? And then our brain can be divided into 4 major regions. This was your first question of your quiz. Okay, so your 4 major regions. The cerebrum is largest part, and we'll talk about him first, but it's all this area right here. That's your cerebrum. In the picture, we have the cerebral hemisphere. So the cerebrum can be cut in half, and you have a right hemisphere and a left hemisphere. But when you put them together, that whole thing is called a cerebrum. The reason they labeled it cerebral hemisphere in the picture is because you're looking at half of it. Okay, you're looking at one half of the cerebrum. Okay, where is the diencephalon? Yeah, he's this big purple thing, and he's deep, like he's in the middle of the cerebrum. You wouldn't be able to see the cerebrum, you wouldn't be able to see the diencephalon if you just looked at the brain. You would have to chop the cerebrum off to see it. Okay, then the brainstem is this green guy, covers all that. And finally, your cerebellum, or your little brain, is this guy in the back. Okay? So hopefully you identified some of those parts on your sheet brain, as well as on your quiz this morning, right? Okay. So we're going to start with the cerebrum because it makes up the majority of your brain. Like we said, there are two cerebral hemispheres, your right and left hemisphere. Down the middle, what separates the two is a longitudinal fissure. Before we get too much farther, did your brains have the dura mater and everything still on them? Yeah, you had to cut that off first? Okay, so cut it off and you look at it, and you pulled the dura mater out, and some of the dura mater came out of that middle longitudinal fissure. Okay, and then you cut through that longitudinal fissure. That very, very middle line. Okay? That's what we're talking about, separating right from left. Okay? The cerebrum constitutes 83% of your brain volume. So it's a lot. A lot of your brain is a cerebrum. And we're going to look at some anatomical divisions on the next slide. Okay. Rose anatomy. So there is a part of the cerebrum called the cerebral cortex. The cortex is a thin layer of gray matter that's all on the surface of the brain. Okay? If it is gray matter, what do we know about it automatically? What does it mean if it's gray matter? So basal nuclei are another example of gray matter. There's no myelin. Okay? So if it's gray matter, that means there's no myelin present. White matter has myelin. So that's the difference between the two. So gray matter has no myelin. If he has no myelin, what part of the neuron are we looking at primarily? Are we looking at cell bodies primarily, or are we looking at axons? What part of the neuron is not myelinated? Cell bodies. So this is going to be made up of cell bodies and possibly some unmyelinated axons. Okay? Alright. So you see in our picture here, down at the bottom that I just drew over, that there is this thin layer of cortex, of gray matter on the outside, and then the white matter is on the inside. So that cortex, that gray matter, is 2 to 3 millimeters thick, so it's pretty thin. But it covers both of the cerebral hemispheres, and it folds on each other. Okay, it folds. So what are the hills of the fold called? The sulcus are the grooves. So the hills are the gyri, or a gyrus is singular. Okay, so the hills are gyrus. And the valleys are a sulcus, or a sulci would be plural. Okay? So we increase the surface area by having these gyri and sulci. Okay, so it gives us more surface area. Even though we're only 2 to 3 millimeters thick, we increase the surface area with those indentations. Okay? And we can separate the cortex guys already know these lobes because you know your skull. So we've got the frontal, we've got the parietal, we've got the occipital, and we've got the temporal. Okay? So separated according to which bone sits on top of it. Okay? Alrighty. Now, some functions of the cerebrum. Now, before we get too far on here, let's talk about this term, contralateral function. So, contralateral function means that the right side of the body is primarily controlled by the left side of your brain, so your left hemisphere. And then the left side of your body is primarily controlled by your right hemisphere. Okay? So, the divisions in your body, there are certain things that are only controlled by the right hemisphere and certain things that are only controlled by the left hemisphere, but then they talk to each other. There's crosstalk between them. Okay? But the contralateral function is right side is controlled by the left brain, left side is controlled by the right brain. Okay, so they crisscross. Okay, so I'll quickly put this up here. You do need to know some of these areas that are in your objectives, but I'm not going to go through and point them out. I'm going to wait until we get to that section on the slide. Okay, so we're talking about the functions. Okay, but big areas to start us with, the central sulcus. Did you see the central sulcus in your sheep brain? Okay, so it should have been a big indentation that's cut transversely across the brain. Okay, when we look at that central sulcus, the things that are in front of that central sulcus are primarily motor areas, and the things that are behind that central sulcus are primarily sensory areas. Okay? So your brain is separated into the motor areas and the sensory areas. Okay? This frontal lobe gets a little weird. He's association areas, but we'll get to him in a second. All right. Erase all. Here we go. Now, as we talk about functions of the brain, let's talk a little bit about how we know these functions. Okay? So nowadays, we can use imaging, like PET scans or CT scans, to kind of see what's going on in the brain, to see what areas are firing up, sorry, MRIs, to see which areas are firing up when you're doing a certain activity. So when you're seeing, when you're looking, you have this region of the brain firing up, hearing around your temporal lobes, thinking is all over the brain, speaking, specialized areas within the brain. So we can pinpoint which areas of the brain are active during certain activities. Okay? Now, that's what we do now that we have fancy machines. But in the development of this science, we didn't have fancy machines, and we had to use other things. So they used human subjects and guinea pigs to find some of these areas. Most of the research was done pretty benignly, where they found a patient who had a specific lesion, and then they looked at the side effects. So if they were impaled with something and part of their brain got messed up, and they lost their memory, they would then go back and say, okay, memory must be associated with that area that was lost. Okay? So they did some brain mapping that way. But, as we know, sometimes scientists cross the line and don't do everything as ethically as they're supposed to. And so there were some scientists who were really, really curious and couldn't contain themselves. And so instead of waiting for a patient to get a specific lesion, they may have caused specific lesions to see what the effects were. So this could be done by somebody who was going in for a brain surgery that they needed, and then they just happened to injure another area at the same time. Or they found patients who couldn't give consent because of disabilities, and then they took advantage of them. So some of that is the history of what we're talking about here. Okay? So it doesn't have a great history, but we can redeem that and use this knowledge to help others, right? All right. So, motor control. So this is going to be the region in front of that central sulcus, okay? The motor control. Voluntary muscle contractions, so moving skeletal muscle, are initiated in the motor association area, so the premotor area. So if we're looking at we are looking at our primary motor area, which is right here in front of it. And then the sulcus, or sorry, the gyrus right in front of that is your motor association area, or your premotor area. Okay? The motor association area. So in that motor association area, that's when you're going to say, hey, I want to pick up that water bottle. What are some of the things I need to do to that? I need to walk over there, and then I need to stick out my arm, and then I need to lift it up, and I need to unscrew the cap, and I need to drink it. Okay, so that's going to happen in that motor association area. You're taking your memories, you're taking your knowledge of how things work, and deciding what to do with it. Then, that information will be sent to your primary motor area, your precentral gyrus, this guy right here with shaded light blue. Okay? That precentral gyrus will then be the one that actually sends the information to those motor neurons, to those muscles, and have them contract. Okay? So motor association area decides what to do. Primary motor area actually does it. Now, you see this pretty picture we have over here of our primary motor cortex, and then we have different regions of the body labeled here. This is a map, a motor map, showing us which areas of the brain control which part of your body. Guys, I think this is one of the coolest things in this section. Okay, so we've labeled it out, and how they figured this out was they would do brain surgery on people while they were awake, which isn't an uncommon thing. You can do that. But they would send in little electrical impulses in different regions of this primary motor area, and they would see which area twitched. So they would stick a little stimulus in your brain, and whoop, the arm jerked. That area must control the arm. And so they mapped it out to see which regions of the brain control which areas of the body. But as you see, it's not all equal. So not every part of the body is represented with an equal amount of the brain. And it's not even size related, because you would think your leg's pretty big. It must be controlled by a big portion of the brain. But if we look at our picture here, what do you see is really big? That hand's pretty big, right? So your hand requires a lot of that motor cortex to control it, because you have a lot of delicate little movements that you can do with your hand. It takes a lot of brain area to figure out what to do with that hand. Okay? You also have, look, there's a whole area for your tongue. Okay? So your tongue, which is super tiny compared to everything else, takes up a lot of your brain. Could be a good thing. Okay? Your face is really big. And then here, like your hip, your knee, your foot, your toes, your genitals are over here. Okay, so smaller as you go around. If we put this on a person, and we make a person with the same ratios as the ratios that we see here in the brain, we get this little guy. Isn't he cute? He's called a homunculus, a tiny person. And he has his body parts are in the same proportion as the areas of the brain that control them. So you see he's got big hands, he's got a big mouth, pretty big eyes, but his arms are really skinny, and his legs are really skinny. Okay? So they don't require a lot of the brain to function. Okay? So, that's motor control. Okay, now, somatic sensation, picking up information and sending it to the brain. Okay? So, first of all, our general senses, like touch and pressure and heat, things like that, are sent to the postcentral gyrus. So it's the primary sensory area, this gyrus right behind the central sulcus. Okay? So it's the primary sensory area, and it has neurons that receive the sensory information from all over the body. Again, we can map it out and see which areas of the body take up the majority of that postcentral gyrus. Okay? And you see it's similar to the motor area, but there's other things that show up here in the sensory area that didn't show up in the motor area. For instance, we have this whole section here for teeth and gums, and jaw, and tongue, and pharynx. Special mouth area takes up a big portion of your brain for sensory information. So a toothache hurts really bad. We think about it a lot because it's taking up a lot of your brain to think about it. Okay? The pharynx, your intra-abdominal area, takes up a big chunk of your brain. So think of having sensation coming back from your abdominal organs. Okay? Your lips are big again, your face, fingers are big. But then look at here, your head is tiny, your trunk, hips, legs, knees, your foot's kind of big. And then we have your genitals back here that are much bigger in sensory than they are in motor. Okay? All right, so sensory information comes in behind the central sulcus. Okay, and then there's just comparing your two homunculus guys. Aren't they cute? Special senses. So these are going to be vision, olfaction, hearing, and gustation. Okay? So bringing these in. So these special senses don't enter the postcentral gyrus. Okay, so the postcentral gyrus is just for general senses. Okay? Instead, they travel to other specialized areas of the brain. So you have, too far. So you have regions of your brain that are specific to each of these senses. Okay, so for instance, for vision, we can look back here in the occipital lobe. We have the primary visual cortex, which is this little light blue area back there. That's going to be the area of your brain that's actually receiving visual information. Yeah. How what? Yeah, so it's shutting down this cortex, shutting down the higher order thinking in your cerebrum, but your lower brain, so your brainstem and your cells may still be working. So that keeps your heart working, keeps your breathing and stuff like that. But your other parts of your brain are turned off. Yeah. And if that happens, brain death is when your brain, your higher order functions have stopped working. So you don't have your reflexes, you're not responding to the world around you. But you may still be breathing and your heart may still be beating because the lower parts of your brainstem and your cerebellum, are still sending out signals to keep you alive. So you have to make the decision, brain death, what do you do? Do you consider that death? Or do you draw the line? So, all right. So there are things set in place where you have a certain number of hours after brain death that you would call them dead versus waiting around to see if they would come back. Okay. So, special senses. So, we said that this back area, the primary visual area, is going to be where the visual information comes in and is received at the brain. But then you see there's this larger area. Okay, that is the visual association area that surrounds it. The visual association area is going to take that visual stimuli and associate it with other things that you know or memories that you have. So that you can look at that water bottle and say, that is a pink water bottle. I know that it is pink because I know the color pink from preschool. And I know it's a water bottle because I've seen a water bottle before. Okay? So you're taking your visual information and deciding what it is based on other things that your brain knows. Okay, so those association areas are pretty big. Okay? So you have, for hearing, your primary auditory cortex, and then an auditory association area that surrounds it. Okay? The primary auditory area is small, the association area is larger around it to help you identify what the sounds are. If you hear a dog barking, you know it's a dog. Okay? Even if you don't see it. Okay, and let's see, taste, you have a gustatory complex, or gustatory cortex, sorry. And for equilibrium and balance, you have a vestibular cortex that are deep within the brain. Okay, so the special senses will travel to a specialized area of the brain, and then the association areas will help you make sense of that special sense. association area or the anterior association area. Okay, this is a generalized association area that takes a while to develop, and you guys, as you are reaching the end of adolescence, are finally beginning to completely develop this area, which is a good thing. So some of the things that are done in this frontal brain area, your working memory rests here. Executive area for task management, being able to use time wisely. Working memory for object recall tasks, solving complex multitask problems, being able to see something and predict what will happen in the future. Okay, so as you are developing this frontal area. Okay, it's really important for decision making. Okay, so it's good that you're reaching the time in your life where the frontal area is becoming developed. Now, if you make bad decisions, you may still have a little wiggle room and say, just because my frontal region is not developed all the way. But you don't have much longer to use that excuse. If you want to use it now. Okay. Cerebral lateralization. Okay, so your right and left hemispheres. We've already said that they do different tasks in your different hemispheres, and it's usually correlated with handedness. So most Americans are right-handed. I know there are some lefties in here, but most of us are right-handed, and most of us have that left hemisphere as our logical or analytical hemisphere. However, if you are left-handed, you have a 50% chance that is flipped. That instead of having a left hemisphere as your logical area, you instead have the right hemisphere as your logical area. Okay? So, because of your handedness, you may have a flipped brain. Now, that probably is why you are left-handed instead of right-handed too. Okay? It's because that logical part of your brain is responsible for helping you determine writing letters and making things make sense. Okay? In general, males show more lateralization than females. So when we talk about lateralization, it's separation of roles into different parts of the brain. So in general, males have things compartmentalized more than females do. Okay? So if you read some marriage books before you get married, it's a good idea to do. But if you read some marriage books that tell you about the way men's brains work and the way women's brains work, you might run across this analogy that female brains are like spaghetti and male brains are more like waffles. Okay, so female brains are connected really well. We have large corpus callosums, which allow our brains to talk from one side to another, and we can think about multiple things at the same time. And we can multitask. And we can go from one thought to another very seamlessly. Okay? Again, this is a generalization. Not every female is this way, and there are females that are this way too, but generalization. However, males' brains are more like waffles that have little squares in them, where they will jump into a little square, and they will sit there, and they will think about what they're thinking about. And then when that is done, they will jump into another square. And they will think about what they think all about next. And then they will jump into another square. Okay, so they can compartmentalize their thoughts a little bit easier. Okay? All right. It used to also be thought that men were better at visual spatial skills, were more right brain, and women were better at the women were better at verbal skills. But a lot of studies have been done on that and said that that isn't really true. Okay? But the right hemisphere is usually associated with those visual spatial skills, intuition, emotion, and artistic skills. Okay. So, some examples. You don't have to know these pictures. You don't have to know what's on here. But just to kind of give you an idea of how the right and left hemispheres of the brain are not identical. They're similar, but they're not identical. Okay, so your right hemisphere, you're going to see memory for shapes, left hand motor control, filling shapes with the left hand. But your left hemisphere, you've got verbal memory, speech, right hand motor control, filling your shapes with the right hand. It's a little bit different in your right and left hemispheres. But again, you don't have to know that. Just understand the concept that things are not identical between the right and left hemisphere. Okay? All right, language. You guys learned about this for your objectives group quiz today. So, language is a super complicated process and involves multiple regions of your brain. So, on the quiz, you just had to list 2 language centers. But let's see what you remember about what happens in each of these. It was just one? Yeah. Okay. You guys have been quiet all class, like not saying anything. But no, it was just one. I apologize. Okay, so let's see if you can remember what these different language areas mean. Okay, so you've got the Wernicke's area. What does the Wernicke's area do? It does what? Yeah, so it's processing and understanding language. So I'm talking to you right now to understand the words that are coming out of my mouth. Okay, that is done by the Wernicke's area. Okay, it's also processing in your brain and knowing what you're going to say. Okay, what about Broca's area? What is it responsible for? Production of speech. So Broca's area is going to be found. Let's see, he's right here, right? Broca's area is up here, more in the motor area. So he's responsible for the motor production of speech. Your Wernicke's area is back here. So it's more about the sensing, the identifying language. Okay? What about the angular gyrus? Huh? Comprehension and comprehension of what? Anybody else got it? So hearing, comprehension of hearing and of reading. So taking language in and comprehending it. Okay, that's angular gyrus. And it's not on the picture, but he's deep back here behind that Wernicke's area. And then finally, the affective language area. What does he do? Do you have a professor who talks like this? Who doesn't change their tone of speech? I don't even know if I can do it. I can't talk about that for too long. But it's just really monotone, and you don't have any inflection in your voice. They don't get louder and softer. They just have like this all the time. Okay? That is the affective language area. So the affective language area gives you the ability to have emotion in your speech, to have inflection, to get excited about things. Okay? So that is your affective language area. Now, that does not mean that you need to go tell your professor who is monotone that they have a problem with their brain, that their affective language area is messed up. Okay, don't do that. You'll get exposed to trouble. Okay. So far, the things that we've talked about have been in the cortex, in that 2 to 4 millimeter thick gray area on the surface of the brain. There's a lot going on in that surface of the brain area, okay? Now, we're going to get to the white matter that's sitting underneath that gray matter, okay? And the white matter is going to consist of myelinated axons, right? Because white matter means it's myelinated, and the part of the neuron that's myelinated is the axon, okay? So these axons are bundled into tracts, and the axons are responsible for carrying those action potentials. So these are tracts where information is going to be carried from one place to another. So we're not necessarily making decisions or causing something to happen. We are just carrying information from the cortex to somewhere else. Okay? Now, there are going to be 3 types of tracts depending on where the information is going. Okay? So our first type of tracts are projection tracts. They're going to be these purpley blue fibers down here that are going like this. Okay? Their job is to take information from the cortex and send it to the lower brain or the cord centers. Okay? So from the cortex to somewhere else. Okay? Or the opposite direction, from somewhere else to the cortex. But it is leaving the cortex. second one, commissural tracts, are going to connect corresponding gray areas of the two hemispheres. So they connect the right area of the cortex to the left area of the cortex, or the left area of the cortex to the right area of the cortex. So it connects the two hemispheres together. So an example of this is your corpus callosum. It's a really thick tract that connects the two sides of your brain. Okay? And then finally, we have association tracts. They are going to connect different parts within the same hemisphere. Okay? So, if you are taking it from the cortex out, it's a projection tract between the right and left, commissural, and then between different regions in the same hemisphere, association tracts. Okay? But the job of tracts are to carry information from one place to another. All right? Okay, now we're getting to those basal nuclei. So the majority of the rest of the cerebrum is that white matter, those tracts connecting things to each other. But you do have these areas that are deep within the cerebrum where you have masses of gray matter. Okay? These masses of gray matter are called basal nuclei. Okay, and they're buried deep within the cerebral hemispheres. Now, you do not have to know the names of the specific basal nuclei. Okay? So the caudate nucleus, the putamen, the globus callosum, you don't need to know those names. You just need to know that collectively they are the basal nuclei. Okay? And they are collections of gray matter deep within the cerebrum. So they are collections of cell bodies that are buried deep within the cerebrum. Okay? The basal nuclei are involved in motor control in the thought process. So, you've got the precentral gyrus that's sending motor control down to the rest of the body. These basal nuclei are what control those thoughts and make them smooth and gentle. Okay, so they're taking motor control and making them smooth. So if you know somebody with Parkinson's disease or Huntington's disease where one of the primary symptoms is you get really shaky, you have problems with these basal nuclei. You still get muscle movement, but it's not smooth at all. All right. Got 2 more minutes. Okay. Our next area is the diencephalon. So we're looking deep in the brain. So if we take off the cortex, what we're going to see is first the thalamus, which is this purple guy right here. Okay, he makes up 80% of the diencephalon, so he's the majority of it. He consists of 2 oval masses of gray matter, so those big purple guys. And they live underneath of each cerebral hemisphere. And then there's a narrow little intermediate mass that connects the 2. Okay, it sits right here and connects the 2. Between the 2 sides, there's a third ventricle, okay, that lives in the middle there. Okay, and then let me just give you the function of the thalamus, and we will be good. Okay, so the thalamus is the gateway to the cerebral cortex. So he's going to take all the information that's headed to the cerebrum and filter it. So everything going to the cerebrum has to come through the thalamus, and everything that the cerebrum sends out has to go through the thalamus, except for sensory input from smell. He goes a little bit differently. Okay, so the thalamus is the gateway. All right, that's as far as we're going to get. We'll pick up here on Wednesday. Remember that you do have a quiz on Wednesday. Make sure that you are prepared for it. Okay? Any questions? All right. Let's see, how far away were we?